I don't know why other people are not joining on time. Because you know that the lecture is strictly for 1 hour, but today I increased the period for 1.5 hour because I think that many students are joining, not joining at time. And you know that in a 50 minutes, 40 minutes, there's too much short time to elaborate and discuss the things. And many students complain that if you do not understand such things because these things are new for you guys. And I advise you join the section and time. And in case during the week you have some still problem regarding the lecture or any other assignment, contact me through mail, with WhatsApp. I am available for us. Ahmed, are you have some query regarding the previous lecturing, a previous working? We are working.
Sorry, I didn't hear you quite correctly. Can you repeat?
I am asking you that you have any query regarding the previous lecture regarding the data pre-processing procedure, because now I am starting the to say that the repetition lecture. Previously, I taught the AI students the different techniques to upload the data MRI in a single view and in the 3D view, and conversion of one format to another format. Now repeat these things and discuss about the scenario, how the data is processed and which steps are performed in the radical format.
I mean, yeah, so I don't really understand what you want from me to do. Yeah, are
Uh,
you asking, do I have a question about the previous topic?
I am previously teaching about the data pre-processing steps. Now
Yes.
repeat such steps. Are you view my screen on your side?
Yeah, I can see it.
Okay. You see that there is a flow chart for the data pre-processing and the model building in the larger abstraction view. First of all, we take adding data set, yeah, another data set in the format of NI. NI mean Nifty format.
Yeah, I
And
remember
then,
that part.
and then we apply the data pre-processing on over Nifty over MRI, because the MRI is in raw format and the skull and other things are already involved in the outer layer of the brain. Because we deal the brain now, we firstly we remove the skull structure. It is the external layer of the in our brain, and then also applying some other data pre-processing like noise removal, voice corrections, and further normalization of the normalization means some techniques apply on that over image. Wait a minute. Manal is also joining us.
Regarding Mustafa, he said he will be like about 5 minutes late.
Yeah, yeah. Assalamu alaikum, Manal. How are you?
Walaikum salam. I'm actually currently outside, but I'll be joining in for a very short period today. I hope that's okay.
Okay.
I will be going through the recorded sessions to
Um,
see what you will be doing today for the whole Zoom.
you are in the traveling? Yeah.
I had family, honestly. So, yeah.
Okay, okay. But I suggest selected some time from the collaboration of all your guys and your concern. Uh, uh, okay,
Yes.
it's fine.
However, the times that we were given were really only on the weekend, which is Friday, Saturday. As you can hear, my baby's here crying, wailing, so it's almost impossible to do anything on the weekend. I have given that suggestion to Ahmed. It's yeah, honestly, really tough to do things on the weekend. But I will be doing the best that I can for the Saturday Zooms. Just give me a second, baby. Okay, Habib. They're currently sick as well, so yeah. Anyway, I will be here for a short period, and I will listen in for however long I can, and then I will just dip out whenever I feel like it's getting a little bit too much, and then I'll go through the Zoom meetings, recordings as Ahmed has promised later.
The recording is also if you have this emergency, and that's why I record or the lecture. And some people are not understanding this thing at the right spot. When we watch the recordings, he understands such things. The main purpose of the recording is that, but the purpose is not that the people are not joining the session and also dealing with the recordings, because recordings are also available in the side of YouTube. Well, the main
It's
purpose
okay.
of
Then don't worry about it. Don't worry about it. I'll just go through whatever you have as if it's a document, whatever it is, I will just go through it myself personally and take it as initiative to make sure to keep up with the rest of the group. Don't worry about
okay,
it. Like even if the recording isn't there, don't worry about it. I'll make sure to get myself sorted.
okay, that's fine. Asma, how are you? Are you fine?
I'm good, thanks. What about you?
And also fine. Now I started the lecture. Now today lecture is the joint section of you guys and some other people guys who are studying or who is practicing in the medical field. Things Faisal Officer Ahmad is also joining us, and two other guys, Mustafa and Abdullah, is also joining us. And a new guy from your side is Khalid. I also sent the link from Khalid, but Khalid still not joining us. In the last lecture, I am telling you about the data pre-processing, how we deal the data. First of all, how we upload the data from Nifty format to uh, in our side, and then some apply some pre-processing. Now the today I am telling you about the which processes are involved from data pre-processing. Especially, I make a flow chart from you guys how the things are doing that. First of all, we upload the data in the format of from ADNI side and some other recognizable sites like OSIS and Parkinson's. And the data is mostly data is available in the format of Nifty. But if the data is not available in the Nifty format, it is available in DICOM. You can convert such data into Nifty format. It's a simple that one line code apply, and the data, all the data is converted to Nifty format. The extension of the Nifty format is N double I, and sometimes the extension is also available in N double I dash GZ. If GZ means the data is also in Nifty format, but in the zip format. You know that zip format extraction format to reduce the size of the data. And then we apply the pre-processing techniques on that. First of all, we apply the skull stripping. Skull stripping means we remove the outer layer of our brain uh, from uh, through some algorithm, and the original view of our brain is come to my side. And then secondly, we apply the noise removal, noise and normalization. Noise means sometime when we perform the MRI from patient, the some images are in the darker form and some images are in the lightened tone. Then we even the tone and remove the noise bit of all the images, and also perform the normalization mean also we perform the size, flipping, rotations, and some other things apply on that. Then all the images are in the same symmetry. Then we apply the segmentation task. Segmentation task is a task we divide our images into split into small segments like tissue segments and other things. That after the all the things we are doing that, then we give to when we maintain a template in MNI format. The MNI is a standardized template format. It is introduced by Monorail Institute, Neuroimaging Institute from Canada. I think Monorail is in Canada. And then we supplies the images in the 3D to 2D format and given to our model, and further the things are doing. I show you some slides, uh, then you better understand the things. Uh, you see my screen?
Yes.
All the person visibility is fine.
Yes.
Okay. First of all, we upload the MRI images, being the original 3D images. As you know that of our MRI in the 3D format, and the x-axis is the cornel view, and spectral view, and third is axial view. And we take all the views of our images, and then firstly apply the intensity normalization. As you see in the images that intensity normalization means some portion are in darker and some portions are in the brighter tone. Then we apply a mask on that, and after the masking we corrected such images in one intensity for the better understanding of our model. And these steps are basically we shuffle these text as to your structure of your model, but basically we prefer that first of all we normalize such images like the intensity normalization. I believe that the structure is better understanding from the better perform of the model. But some people do that, they firstly apply the skull stripping, and secondly intensity, and then registration, and after the tissue segmentation, and then at last any data accumulation. And you, it's your own which thing is more feasible to you, and which thing you believe that these process, yeah, these steps are better for your model tuning and better result predictions. In the second thing, we apply the registrations. Yeah, I go to the slide. The registration is also available. Registration means uh, we apply, we made a template mean you see that when you capture snapshots from the cameras which are not digital, and these are in the real format, all the images reels are in the form of a template, in one format, in one size. Similar structure is applied on such images. We apply made a template. The template is MNI. MNI is a format of Montreal Neuroimaging Institute created by the such template. And I selected 152 means around about 150 images are available in such fit in such template. And after fitting such images, you see that all the images are same like in hierarchy, but the nature of the images are different, and the skull is also in the outer layer of the image. uh, it's your thing. You made a template before skull stripping, yeah, you made a template before after skull stripping, it's depend on you. And after that we apply skull stripping. This step is a major step because after before applying the skull scripting, we cannot analyze the basic structure uh, of the brain three tier structure. You see that this is a three tier structure. This is the axial view, and this is a spatial view, and this is a coordinate view. The three views are available for us. And first of all, we upload the image in T1 weighted format and apply some bias correction. Wait a minute. The bias correction I apply such like that. You see that our skull is in the 3D format. Uh, I think someone will join us. Wait. Manal is also leaving us. Asma, you are available, and Zian is available. Dua is not available. I don't know why he's not available. Now continue that. Skull stripping is a process which we remove the outer external layer of our brain. You see that there are three D format of our brain. First is the axial view. Axial is a Z format. Z axis. You see that this is Z axis. We rotate all the three view of our brain. And the corner view is a horizontal view. You see that this is a horizontal view and this is a corner view. And sagittal view is a y-axis view. You see that this is the y-axis of our area, and this is that. After we uploading the MRI, we see such three type of views in our site. Ahmed, are you see the MRI in the real form?
Only pictures.
Only pictures. Now I provide you some demonstration and some codes. I think that you know also about the Python, and you do that on the Colab.
Yes, I'm familiar with
Okay.
it.
Okay. Now I repeat the code. We repeated the last lecture because many people do not understand that the things, how the things are doing that. Because these things are new from you guys, and I am doing a lot of here from such programming like stuff, and in the neuro programming, I think that I am working for last 7 to 8 months on such stuff, and also researching more things that because it is a very deep research field, not an easy task, and I think that it's more complicated than the other part of our body. Ahmed, I am right saying that?
Mostly, yeah.
Because after the brain, I think that the heart is a more complicated because many veins and many other things are involved in that. The surgery and the diagnostics of the thing is not easy. Um Heart is too much not complicated because many things are also available that many. But I think that the only way you diagnose your brain through MRI and fMRI. Now we have three view: axial view, sagittal view, and corner view. When we upload our MRI, all the three views are available from on site, and these are in the form of slices. When we move through the slices, the different angles and different slices are moving through from on the screen. I present you and demonstrate in the my side after the presentation, and you see that when we remove the skull, these images are all available in the form of skull. This is skull, this is also available skull, and this also is skull. And you see that all the images in three views are skull. But after removing the skull, you see that in corner view, when we remove the skull, the real form of our brain narrow image. And secondly, when we view on the sagittal view, you see that after the skull, the view is that. And the third view is the axial view. Normally, the doctors diagnostic more things from the axial view, Z view, because the Z view is most clerically view, and the tissues and other things are also visible in that type. And you see that we have three type of it's a black and white image. We see that we have gray area and white area and some flooding area like that. Now we move to the other step, and other step is that tissue segmentation. Somebody join us. Mustafa is also join us. Assalamu alaikum, Mustafa. How are you? Assalamu alaikum, Mustafa. How are you?
Alaikum salam wa rahmatullah. I'm good.
Mustafa, you are late for 30 minutes. Please maintain your timing. You join the session
I'm,
right time because
I'm
you
sorry.
know that. No, it's okay. I know that you have a busy schedule because you are uh, something tough scheduling. But to manage that, you join the session right time because many things goes forward and we move backward to explain such things. Are you understand my case? Now we go to the most important. I think that after the skull, this is the most important part of our data processing. It's the part of tissue segmentation. The tissue segmentation, you see that uh, it's our main MRI and also a fMRI. Then we labeling our MRI and fMRI, and CSF is a cortical island fluid, serial fluid, and GM mean gray matter and W mean white matter. We also extract all such things from one image, like these images. This is our axial view, and you see that gray area, the white area, and this is a CFF area, fluidish area. Mean some like you do not add the fluid area or in our blood and in our veins, such area. And after we applying some tissue segmentation algorithm, we coloring such areas. You see that we give the reddish area to our white area, and the light gray area we also implement to the yellowish area, and the fluidish area we represent to in the blue. You also apply different type of color scheme as you wish, because every person applied different gradient uh, theme on that. And in this picture, the red, blue, yellow uh, RBGC coloring scheme also apply in that. And after that, you see that we extract our brain MRI, brain extraction, and labeling the that label means different parts labeling, like these are these tissue, these are tissue anatomy labeling that for our feature extractions. And after apply that, we distinguish which is the gray matter and which is the white matter area. When we apply the data extraction streak, we more deeply know about that, how these things are working. Now, in the last segment is the data augmentation. Data augmentation mean that when we give the input in the MRI and fMRI format, firstly, we convert all the images in 2D slices because we cannot apply the things on 3D slices. It is very clear view for all of that. The doctors see the images in the 2D slices format, but MRI in the 3D format. When we are dealing to fMRI, the fMRI is 4D format, 3D as a imaging format, and 1D is time series. I When we goes to move forward to our programming task, I move through a demonstration example of MRI and what is the difference between fMRI and MRI, because MRI is a simple thing and many doctors also working on MRI because fMRI is so tricky and too much difficult because time series and other bold analysis, blood oxygen, liquid dimension analysis is also involved that. Uh, or you have some question from last slides, then I concatenate all the things. Asma, you have some question? Asma, are you
Hello,
here?
Yes.
Ahmad, you have some question?
No, everything clear.
And Zian, you have question?
No, all is well.
Okay. And Mustafa, you have any question?
No, thank you, but I'm gonna watch the recording. Where can I get the link again?
I am giving you all the recording and share you a YouTube link. I prepare you for a YouTube channel where all the recording in the format of playlist are also available. And I am also sharing you previously a GitHub repository like that. For you people, I prepare a Neurocogni AI repository and which repository is also available NCAI training program. In this such repository, all the week one slides and other materials are also available. That you see that, and in week two, the also we taught the that different type of codes and also the data sets are also available in that in the MRI and fMRI format also available in that. Okay, data
Okay,
slices
thank you.
are
Thank you.
the today lectures also slides and other things are also uploaded in such sites. And still you have some query regarding for the understanding of such thing, you may uh, email me or through other source like WhatsApp. You contact me, and I try my best to give you best resolution of the query. Now I am telling you that the last process is that in the data pre-processing is a data augmentation. Data augmentation is normally not a hard task because such things are also doing you that in different like of tools for the formatting, cropping, and other things like that. You see that some images are in horizontal view, some images are in the vertical view. Then we rotate some images like flipping x-axis, flipping y-axis, and other thing is that transitions of images and sharing and scaling of images. Scaling means that we make a one size of all the images. Normally we have dealing that 250 by 50, 170 by 220, and multiply by 270 3D like that. And also the noise in blurriness and brightness also covering in such site. And these are the uh, just like coding steps, which we zoom the images, rotate the images, just like giving the methods are also available, libraries are also available. Kera also providing you the data pre-processing library, TensorFlow also giving you the facility, and some also NI libraries. NI mean narrow imaging libraries like NiBible and other libraries are also providing you such methods to just give the parameters and the values in such parameters, and all the things are doing in the symmetrical way in the batch format. That now we go to this is a single multi classifier. If we apply a classifier means we predict a disease category. Now the process is that we are giving the data. After giving the data is preprocesses in the 2D format image slices, and 2D image slices are providing to our CNN network. CNN means convolutional neural network. And after applying, when we make a model, the other things we are explaining you that how we develop the model and what things are available in the convolutional neural network, which area is training, which area is testing, and which area is validation. After we train the model, I am giving a nifty format MRI or 2D slices to our model, and our model predict that which category is Alzheimer or such other like disease. Let's give the example for you. If developing a model of a tumor of brain, you see that I researched that there are more than 44 type of tumor categories in a brain, and simple is that seven categorize, we simply categorize such categories In the larger view, there are, I think so, the 7 classes are available for the brain tumor, and we train our model all the brain tumor images slices through MRI. And when we give our fMRI slices to our model rightly predict that which category of tumor, and also with the help of explanation AI, pointed out the area. This is the advanced level of modeling. Also explain that such portions are affected for tumor, and that's why this images relies to such category. This is advanced format. I also discuss the format for you. Now I am giving you some other example flowchart. If we made a multi-level classifier is that you see that I take 2D images of one view. I mean that I choose in last model, I choose one type of data like Excel data set. And we take Excel view. Are you, Asma, understand? I am saying that, Cecil. I am explaining also such thing in the last model. I repeat that I have 3 view of our pane view. In the last model, I am developing a model from one view, but in the larger perspective, we are making a model in the all three type of view. First is axial, cornell, and secretarial view, and all three types we made a convolutional neural network. And after the classification, yeah, the prediction of all the such model, you see that if our axial view point out that the person has Alzheimer, and sagittal view also pointed that the person has Alzheimer, and cornell view also pointed that, then I can categorize that the person finally such type of Alzheimer such person has. But in case over because we get a major instance, if over 2 views like axial and sagittal view pointed at that the person has Alzheimer in the moderate brain dementia, and the corner view is not justify such thing, but the majority of the voting is two side now, which suggests that predict that the person has a moderate level of dementia. If somebody explains such model, Zian, all you explain that such model. I am explaining you one by one. Some all the in your own wording, or you understand that how we are telling you how the things are doing that. First of all, Zian, are you here?
Yes. Yes.
You elaborate such thing that how the model is going on. First step is adding data set. Now move on.
I would summarize it as it goes through through a certain process where I'm not sure how to explain it since I haven't got the exact idea, but it goes through a process in order to differentiate or diagnose the La ilaha illallah, the to
No,
like the
no.
patient what does
I
he have.
I I
And,
I I help you that. First of all, the first step of our this is a flow chart. In the first step of the flow chart, we are giving the data set. Data set in the adding and some other format in the format of MRI. Okay.
yeah.
The second step is pre-processing step. You see that such area is pre-processing, and pre-processing also doing such steps like skull stripping, bias correction, noise removal, segmentation, such like that. After
Yes,
the that,
the skull stripping is like to like see the skull from different angles and remove certain
brain external part, we remove the skulls and get the brain from the skulls. This is a
yeah.
skull stripping. And bias correction is that we maintain the intensity of our images, because some images are darker tone and some images are lighter tone. We make the one tone from the intensity because maintain the homogeneity of the images and clarify that the integrity of the images. And the other thing is that segmentation. Segmentation means we cut our images for different segments, like tissue segments and some white areas, some tissues, some other areas, just like that. And after that, we when we completed the pre-processing the section, we provide the data in the 2D format to different all three views to our model and made three models. First model, we create the Excel dataset. This model is called Axial CNN model. Second is corneal dataset, and this is we call corneal CNN model. And third is sagittal dataset, and we more sagittal CNN model. And after get the predictions of all the model, we made a combined classifier, and the classifier giving me results. This result is an accurate result. You see that you say that we made a hybrid approach of all the three models. This is a more authentic approach, as soon that we tell you the previous as a single model is a single classifier model. All the people clear such things? Ahmad,
Okay.
are you clear?
Okay, so does every image
Yes,
or
I'm here.
2D image go through these three processes, or
Yeah,
does it
yeah.
go through one?
In one our actual view, I repeat you like that, you better understand that we have three view. You see that first view is corner view. We get the all the corner slices, and after the skull removing, we all the corner slices made a one model. This model we call axial view CNN model. And the second is that view is that sagittal view. We remove all the skulls, all the data processing on that, and get the sagittal 2D slices of such model. And similarly, the third one that. And after giving, if you see that the first model giving me the prediction that the person has Alzheimer, I assume that. And the last image also giving the prediction that the person has the Alzheimer. Now we predict that the person has Alzheimer because the majority of two images views giving me the prediction that the person has Alzheimer. And the third view is not clear. Just like your idea, we developing a model, because in the next session we also developing a model. Now we move to the demonstrations of such things. In the last session we also demonstrate you people that because many people are new now, I also demonstrate you such things like that. First of all, I give you take a MRI. Uh, this is a MRI subject1.ni file. This is available on our dataset. You see that in our dataset, the MRI Nifty and two subjects MRI are also available in my side, subject number 1 and subject number 2. Okay. First of all, we install three libraries. First library is NI Bible. NI means all libraries which are starting with NI. It's mean narrow imaging library. Or you must understand that when you see some package or some library in the internet and start from NI, you must understand that the library is neuroimaging library. And the library which start by from PY, you also understand that this is Python library, because this is the abbreviations of some people to understand which type of libraries involved in such package. Uh, and the cycling library is NumPy library. Uh, Asma, I think yeah, too, you also know that the NumPy library is simple library. Azyan, you know that the NumPy library?
NumPy?
NumPy, NUMPY. This is a dataset library which is available from you people. And I also giving you some YouTube notes, basic understanding of the data analytics. And in the next lecture, I also demonstrate you such libraries, basic libraries, because these are too much initial things. I am not giving you and wasting such time to such libraries, because we are dealing some high level things, and I believe that mostly people are also in the AI field, and AI field, this is just like ABC of the AI, especially the Python.
I have
And
downloaded. I have downloaded all the except the all three files you uploaded on GitHub, and I have them right now.
are you all people? Asma, Dua is not available. Manal is also lefting, but he said that um, he demonstrated and he tested all the our program in our site. Asma, are you tested all the program in your site, which I shared the code in the GitHub library? Asma, are you here?
Yes, I'm here. Yes, I used this library. It's for the image of MRI and CT scan.
This is Alzheimer images.
Yes.
These all MRI I am taking from admin side, and these all MRI are belong to the Alzheimer disease. Okay. First is library. First, the MRI is that the subject one. Uh, you all, the assignment is you all people that Ahmed and Mustafa, you also try that. If you have some problem regarding the performance install this task, I also taught you giving you some extra meeting and extra high tech class regarding such, because you are doing so not directly dealing with the programming task, but try yourself that you demonstrate, yeah, you run such code on your side. Okay. Mustafa, are you here?
Yes, I am.
Okay, I am telling you that you download all the source codes, and if you have not a Visual Source Code environment, you just go to the Colab and install such libraries. First of all, you install pip ni bible, and secondly you install numpy, and secondly third is matplotlib, and you all copy the code. Yeah, open the such file code in your Colab and run that, just like that. You run the code in your environment like that, and after running the code, you see that the code is running, and after running that, you see that 150 view of x-axis view is available for our MRI. This is a x-axis view. Now we select 140 slide, and view is that y-axis view. You see that this is a y-axis view. Now we move to z-axis view, 140 z-axis view. The first column represent to x-axis, next column represent to y-axis, and third represent to z-axis. This is a simple code. I explained you that we also declare a variable. You also name that as your feasibility, like that image load, other load. I name that test load, and after that, in nib library, I call a method which is load method. The load method demand me that the path of our file. We give the path that dataset. You see that dataset, MRI Nifty, and subject one NI file. And after giving the part, dot get data is a function which get the image from such all the images from the MRI. Okay. Now we load the images in the type of shape because our images are also in the form of pixel. Now we regenerate the pixel to shape, and also shape we are we give the three key view of our slice. First is array is that over x-axis, second is y-axis, and third is z-axis. And after that, PLT is a matplotlib library which shows the images in the plot format, in the graph format. We give the instance of a PLT, and after PLT we make a method of inshow and in give the instance of the test, and after that we show such image from our side. This is our simple code to a 3-line code which we give the MRI, one patient MRI, and we also get the view of the MRI in different angles. Are you have somebody have a question regarding that?
No, thank you. But we can find the data set in the GitHub, huh?
No. I also provide you data set. You don't need to get the data set. The example data sets is also available on the repository. You just download the repository and test in your site from Colab, Google Colab.
Okay.
Are you using Google Colab?
I didn't use it before, but yes, I could use it.
No,
Yeah.
no, no. I give you example that you just go to the Google, because the session demonstrating you to both the people guys that both the things are similar to both Colab. You go to the Colab, go to the Google Colab. After you go that, a Jupyter notebook is also available in your site. This is an online version of the Jupyter notebook. You also go to new notebook. When you click on the new notebook, you see that a new notebook is available on your site. I also run the code in Google Colab for you people because you understand better that. You see that this is a code view also available in your site. I'll just copy such libraries, such code, and paste it in the Google Colab. Because most such libraries are also installed in Google Colab site. If this is not available, then also install such library inside. You see that it gave a error. The error is that NI Bible is not available inside. No. The error is that the subject such data set is not on in your site. Now I give you example on that how you upload the data inside in the written format because time is too short and we are not creating the instance of that uh, in such upload. Wait a minute. You see that the Nifty format is not spotted in that. Now we are uploading the data in some other format. Upload. Now the file is uploading that. I think the file is uploaded. The file is uploaded and file is uploaded under the sample data. Now we take, I think so there is a path setting of that. contents, content, and content sample underscore data. This still the path error. Now I give you example right path of that, because in Google Upload you also demonstrate the path uh, and the path hierarchy is also. Wait a minute, I mount that connected drive. Now I am sending you the file,
Maybe
which all the path structure is also available, because this tool take some time for path structure, and path structure is also setting in the Google Colab. I am setting you the tutorial format how you upload the files in the such structure. Now we move on. The very important thing is that we plot such axis in 3D format like that. You see that I also upload the Nifty NI image, and I give the all three views for our in a slider format. You see that I run such code. Code is running. You see that a slider is also available on that on 3 slides, and I just if I give you doctor in the form of the tool, and you people also make such tool and also upload it in the standard GUI format for your study point of view and for your portfolio. You see that we move to the slided. The images are also changed in secretarial slides. You see that in a 122 image is more prominent, that such like that if the Excel goes to Excel view, we move to the 142 slide, just like a play and plugin tool, which are also providing you doctors for your A's, and you upload any type of library MRI, and the MRI personal MRI are available on the screen for you with different analysis. Is it clear for you people?
Yes.
yes,
Okay,
thank you.
okay. You have any question now? Then I think that it's a 1.5 hour lecture. If you have a question regarding that, you ask me, or if you have some problem from the understanding, you watch such video and note down your question, and also sending me in the format of email, or yeah, some other format like WhatsApp. I prefer you send me such things through email, because in email all the things are in a formal form.
Sir,
Yeah.
I've got a question. Will you please go back to the last file you were demonstrating?
Yeah.
Okay, so in the MRI Nifty file, I've downloaded it and I have it in my
Yeah.
Visual
Yeah.
Studio
Yeah.
Code.
I explain you such code. I first of all I make a method from loading of the Nifty file. This is a method of load Nifty. Okay. Such method is that we give a file path to such method, and such method returns me a data and the loading of such method. And second is that we make a method of orthogonal view. Such orthogonal view is a 3 axis view of our method: x-axis, y-axis, and z-axis. All the axis code is available is that x-axis, corneal, and the last we just plot such view and show such view. And the other thing is that we made an interactive view. This is an interactive view. You see that teach such views as interactive view. In interactive view, we provide you 3 sliders: corner view slider, Excel view, and segmental view. Now, are you understand such thing?
Kind of, yeah.
But you just understand such thing that in a deeply prospective, I also demonstrate you when we giving you some other examples and some other demonstration about the MRI programming. You better understand that. First of all, the main thing is that I am giving you all the people such demonstration. You all people just like run such code and understand such thing how the things are going doing that on your system, on your environment. And I believe that in next lecture, the all people do such thing on your side. Okay.
Okay. Sir, can we, for example, if you have any free time, like check if we have all the files uploaded? Like we can
Yeah,
share
yeah,
also.
yeah. You shared me that through email. I am checking that.
Okay, amazing. Thank you so much.
Okay, Asma, you have some question regarding that?
No, I don't have any question, but I created some code, my own code, and I get the result. But when I used your code, there is a lot of error. I will email you about that.
Yeah, you share your code for me. I check that code and I guide you that which thing are doing you good at and some improvement in your code. I also pointed that you and comment that these things are also improving that way. And Ahmed and Muswa, in your side, you just go to, I also email you the repository. You just go to that and download the repository. And after pasting the code on the Colab, I also giving you the example how the path setting on the Colab, and you just run such code on the Colab. And in the next lecture, um I you must try yourself. You do many uh, something like 1 or 2 code easily you run on your side.
All right. Okay. We'll do so.
Okay. Okay, now I think so. Many things are new, and you cannot understand in deep level. But I believe that if you try yourself and make a concentration and some motivation on that, I think that you all will be a good coder. In the next lecture, some improvement. I believe that improvement on your side. Okay, thanks, and have a nice day from. Okay, Allah Hafiz.
Allah Hafiz.
Allah Hafiz.